Good afternoon, everybody. I'm talking, changing tracks a little bit and talking a bit more about the host and a little bit too about some of what we've been doing the last few months, which is still very preliminary at this stage. So we're very interested, our group, in looking at immune memory to SARS. And I guess for those that are perhaps outside of the memory part, following viral infection, you'll usually generate a very specialized cell subset called memory cells. And these are really important and are what allow you to basically protect yourself against reinfection. And so these are generally classified into sort of your CD4 T cells, which are like the conductor. They're sort of very important for leading the immune response against the infection. You have your CD8 T cells, which are really very important in killing cells that are infected and really active during infection, and B cells that are really have a number of different roles, but one of the most important is to make antibodies that help mop up free virus as it floats around. That's an oversimplification, but that is the sort of the general tract. So what happens is once you get infected, you will hopefully generate a nice memory response once you resolve that infection. But over time, that would generally decline. And this, I think, is one of the things that now we really want to try and understand with SARS, as most of the world really gets control of the SARS-2 spread. And obviously at this stage, we don't really know what's going to happen, whether there'll be a sort of a second surge or another wave or what will happen. And so I think immune memory is very important. And we also don't know, I guess, the importance or the effect of the vaccines. So in terms of immune memory and their durability, it really varies a lot for different pathogens. This bar graph just shows sort of the immune memory against a number of different vaccines, against a number of different pathogens. And so some of the best ones like vaccinia virus can be up to 50 years, but some others like whooping cough are much shorter and require boosting. And there's a number of different reasons as to why memory might vary between pathogens. But what we really don't know is about SARS and what it looks like. So what has been a lot has been starting to come out now, though, is what does immune response look like during acute infection? And this is a really nice paper that's just come out in Nature Medicine that's done some transcriptomic work on the total immune cell population. And so it's a nice UMAP of all the different sort of immune subsets that are there in circulation. And these are mainly in the bronchial lavage sort of samples. And what's really coming out from some of this data is that during an acute phase, there's this skewing towards sort of these severe cases having a much higher number of macrophages infiltrating into the lungs, where a lot of these other sort of the white blood or sort of the memory part of the immune cells are really being reduced in severe infection. This is often referred to as lymphopenia. And so it'd be quite interesting to know post-infection, how well does this memory is able to establish itself given this sort of skewing towards reduced lymphocyte numbers in acute phase. We're also seeing very much a difference in terms of the phenotype of the immune cells that are present during mild and severe infection. So mild infection, we're seeing a really robust immune response, is polyfunctional, sort of CD8 T cells and CD4, and they actually also have a higher amount of this subset called Tregs, and Tregs are very, very important for controlling a lot of things like autoimmunity and dampening an overactive immune response. Whereas in this severe infection, we're seeing a lot less Tregs, which goes along with the fact that there's this over sort of pro-inflammatory response during the acute phase, and also this sort of loss of polyfunctionality in these sort of CD8 and CD4 subsets. And so it's sort of becoming clear, I guess, some markers in the immune cell markers for severe infection. And these sort of markers suggest that there could be a difference in how the immune response develops against this infection. And so I guess one of the big questions that I've said is protective immunity being generated against SARS. Now the data coming out is quite varied in terms of who seroconverts. So when I refer to seroconversion, this is who makes an antibody response against SARS-2. And I think the earlier assays were suggesting there probably wasn't very good seroconversion, but I think we do have to sort of wonder about the quality of some of those assays that were used earlier on in the outbreak. Now I think the data is suggesting that probably 80% or more are seroconverting to the infection. And this is important to know because, as John mentioned early on, and I think some of the others, about plans now to really understand serosurveillance. So using these antibody titers to actually work out the infected population size. So it is important for us to know the seroconversion rates in populations and even in population groups. So, you know, the elderly, the children, and also those that were quite sick as well. So if we have a look at sort of immunity that was generated in the other common cold coronaviruses, we see that T cells are reasonably long-lived, sort of 6 to 8 years. So this is on the shorter side, I think, to many other pathogens like vaccinia and hepatitis B vaccines, but they're sort of longer compared to the B cells. The B cells are actually thought to be very short-lived, perhaps even a few months, and not be that long-lived. And the antibody titer variation for the common cold coronaviruses and also SARS-1 really does vary quite a lot between the different studies. And I think this is quite interesting, and I think it's very important to understand this. And I think one of the things as well is that John mentioned that corticosteroids are probably going to be used for a lot of the treatment, and it has been shown that corticosteroids in SARS-1 can actually reduce the antibody duration. So I think we do need to understand antibody titers, both for sort of surveillance studies, but also for understanding, you know, if the virus was to come back around again next winter, which some people are speculating, will those that were infected in this sort of winter period be protected, or are they susceptible and should they be considered for vaccination? And I think the fact that the B cells are so short-lived, because they would normally go on and generate more antibodies, is a bit of concern. And so we are hoping to try and answer some of these questions. So we want to understand, is protective immunity generated? Does the duration of the immunity vary between the patient groups? And so I think this is, you know, the young, so children, adults, the elderly, and I guess, you know, severely ill versus mildly ill, I think are the sort of the main groups. And also, how does treatment perhaps influence this as well? So in particular with the severe infection is the lymphopenia, which is basically the loss of a lot of these lymphocytes, which are the progenitors for immune memory. Is this going to compromise the immune memory that gets established? And also, does this protect against reinfection, or what do we need to protect against reinfection? I think is still unknown, very much so. So to be able to try and answer some of these questions, what we've been very busily doing for the last few months is establishing a natural history cohort. So we're basically trying to enrol across 8 different sites across New South Wales. And this cohort is called COSIN, is for the adults, and COSIN K is for the kids. But basically we're trying to recruit anyone over the age of 3 years old that was diagnosed with SARS-2. And the plan is that we follow them periodically over up to 2 years. So there's an acute, sort of a more intense acute phase, and then we sort of follow them sort of every sort of 6 months or so, up to 2 years. And we collect both clinical information, so the severity of their illness, their treatment, and a lot of that sort of information, and as well as we're collecting some specimens as well. So we're collecting serum to allow serological studies, but also some EDTA and PAX genes to allow study of the immune system, so the lymphocytes, immune cells, but also to facilitate some PAX RNA transcriptomics studies. And I guess one of the main points to point out with this cohort is that it is being established to be accessible and to be able, for many different researchers across Australia, to be able to apply and use these samples. So far we've enrolled 50 patients at the 1 month time point, and over the next couple of months we're hoping to enroll over 200 patients with follow-up up to 2 years. So we're still in a rush getting organised with the governance structure, but we are hoping that people can be able to apply and have access to specimens to do research related to SARS-2. So it's there to facilitate research. So I guess a little bit, because this is a genomics group, I thought I'd mention a little bit on what our plans are, we're hoping to do. And so we've got different groups that we're going to examine, so ill and mild ill patients, we've got children, adults, and the elderly, and we really want to understand the longevity of the neutralising antibodies over time, the memory B cells, the CD4 and CD8 T cells. And I'll just have a little bit of an overview how we plan on doing this. So we're in a PC3 facility at the Kirby Institute, so we are able to actually grow the live virus, luckily. And we also, though, have generated a sort of a fake virus, what's often referred to as a pseudoparticle, which just contains that S protein, the spike protein, out on the surface. And that one we can utilise in PC2. So we've got those established. And this is just really a nice photo of some nasopharyngeal primary cells that have been infected with SARS-2. And on the left, you can see that's just no virus, that's what the cells look like. And in the middle one, you can see that the cells have been infected with the virus, and they just form these giant cinctia. So you can see the loss of the membrane structure and all the nuclei forming together. And then just the one on the right is an antiviral component that we added. It's called antiviral X at the moment to protect the compound, but it worked very well in cell culture. And you can see that it did confer protection against virus replication. So we're going to use a number of these different systems to measure neutralization and how long it's maintained over time. But I guess if we move more to the exciting genomic component, I guess, of our plans, is that we're currently generating and developing some tetramers. So these are, the spike protein is made as a recombinant protein, but we're also making some CD8 tetramers as well, which are the sort of the CD8 epitopes that are presented as proteins, and then they're bound to the memory cells that are in circulation in the blood. And then we're able to sort these out via flow cytometry, and then we can group these together. And one of the methods we're thinking of trying to do at the moment is to try the BD Rhapsody combined with the BD AbSeq oligos. And we've thought to try and give this system a go, which will allow us to basically look to quantify the protein expression on the surface of the cell. It'll also be able to tell us which tetramer they're binding to, and it'll also then allow us also look inside the transcriptomic data. And a lot of this work has been done that I'm doing is with Fabio Luciani as well, who's a very good bioinformatician and T-cell immunologist. And BD have a number of different antibodies that we're using. So it allows sort of higher dimensionality really than the standard flow, perhaps not as much as some other methods, but I think the fact that you can combine this with the transcriptomics does make it quite a powerful tool to sort of have the proteomic and the transcriptomic combined together in that system. And we'll also be planning to reconstruct the VDJ sequence as well. So this is the immune receptor that sits either inside the T cell or the B cell, and allow us to measure the sort of the complexity or the diversity of that immune repertoire that is reconstituted afterwards, because it's possible with the lymphopenia that they might have quite a restricted T cell or B cell receptor repertoire compared to those that have more mild infection. And so the diversity of that repertoire, it probably indicates better protection against reinfection and re-exposure. So I think they're most of our plans. I think that the most important part is really the cohort that we've set up currently, and we're hoping to really facilitate some very powerful SARS-2 research, and just, I guess, some of our particular plans about our group and where we're heading. So there's a number of people really that need to be thanked. The COSIN cohort it's been challenging to set up initially at the start of the outbreak, and it's a number of excellent clinicians across a number of sites that have really facilitated this process. And we haven't had funding, so we've had a lot of in-kind support from New South Wales Health Pathology and from the hospital sites as well. So it's really kudos to a lot of them that we've been able to set up this, and hopefully we can do something great. And I think our group is mainly sort of Professor Luciani's the main person that I'm working with to sort of facilitate our own research. So I think that's most of what I was going to cover, and I think I'll stop sharing. So thank you.